Thank you for the introduction. Good morning, everyone. I'm very glad to be here. Today's my presentation is about MALDI-TOF MS project. I have no financial relationships to disclose. The opinions expressed are those of the presented and do not reflect those of the ILSI. This is a list of important emerging issues surrounding Japan. For example, increase in microbiological risk in food chain due to global warming, increasing expectation that functional foods will extend healthy lifespan, and creation of foods with novel and high functionalities generated by progress in food science, and pursuit of higher food safety and reliability in Japanese society. It means zero risk belief. And last, expansion of a strong request of animal testing ban from NGO to a food industry. We are challenging by scientific approach to address issues. For example, further progressing in risk assessment technologies, we are deepening precise technologies for microbiological risk assessment and development of new strategies and technologies for assuring safety of novel functional foods, development of non-animal testing methodologies for safety evaluation of foods. This is our task force objectives. The task force aims to gather and research scientific information on food microorganisms, especially harmful microbes, that is useful to the food products industry from a broad perspective. It will also disseminate the results to all the food safety stakeholders through publication, symposium, and lectures under the name of ILSI Japan. This is activities of Microbiological Food Safety Task Force in ILSI Japan. First, development of a rapid identification method for food bacteria and molds, MALDI-TOF MS projects in this presentation. Second, harmonization of inspection methods of spore-forming bacteria. Third, information gathering of microbiological risk of chilled distribution foods. Fourth, reduction of consumer complaints of PET bottle beverage after opening. Final, NGS Next Generation Sequencing Project, collaboration with ILSI Europe. MALDI-TOF MS project's background. Spoilage of food products due to microorganisms is one of the major problems in Japan. Information on the safety risk should be obtained as soon as possible when the spoilage occurs. Identification of the spoilage microbes is extremely essential, but current methods for mold identification are very time-consuming, hours to days. Our goal to develop a novel rapid technique for identification of food bacteria and molds and verify the standard protocol. What is MALDI-TOF MS? It means matrix-assisted laser desorption ionization time-of-flight mass spectrometry. This technique is based on fingerprinting. A fingerprint of molecules composed mainly of microorganism-specific ribosomal protein is obtained by MS, followed by searching and matching with a known microbial strain library and identifying the target bacterial species. If we use this technique, bacterial identification will be drastically quick. This slide is principle of MALDI-TOF MS. Analytes are mixed with biopolymer, matrix solution, and ionized by strong laser irradiation on the stainless steel plate. The ions generated by the irradiation are analyzed by mass spectrometry based on time of flight of the ions. Actually, we are using DNA sequencing for identification. This technique is gold standard method, but it requires scientific background and needs 3 to 4 hours. But by protein fingerprinting, identification results we get in minutes at very, very low cost. This technique is based on fingerprinting, so microbial strain library is very important for fingerprinting with molecules composed mainly of protein. But this technique started in the clinical field. The library included mainly clinical bacteria. So we are Japanese food industry, so nobody had tried to expand the library for food industry. So we task force, microbiological food safety task force as one, made much effort to combine the equipment manufacturers to expand the library for spoilage bacteria in food industry, and provided them with non-microbial strains owned by Japan Canners Association, JCA. This is an example list of food beverage spoilage microbes. This list is yeast. Species with we want to control with high attentions are highlighted with symbols red point. Molds lists. Spore-forming bacteria lists. This is Alicyclobacillus list. So we provided the equipment manufacturers with a list of various food beverage spoilage microbes needed in food industry, but it is time consuming to expand the library in only their efforts. So therefore, we decided to move forward to the more practical collaboration with JCA. JCA has an in-house culture collection of wild spoilage strains from varieties of canned and lettered food in the history. It amounts to 340 strains in total, including 178 strains of aerobic spore formers, such as Bacillus, 94 strains of anaerobic spore formers, such as Clostridium, and 68 strains of thermophilic anaerobic spore formers. But since their collections are mainly characterized physiologically in the past, so they agreed to share that strains with ILSI Japan and MALDI-TOF MS manufacturers for the purpose of sequencing and MALDI-TOF MS database establishment. 16 years rDNA sequencing was carried out on the entire collection of JCA. Identification was based on the upper 500 base pair of 16S rDNA, while the full sequence was applied if the result was not decisive according to the international clinical and laboratory standards. This is an example list of re-identification. For example, pyrimethyl polymyxa were re-identified to different name species. So, and full sequencing results for identification of strains of JCA culture collection were registered to NCBI Gene Bank. This is the expected number of new strains and species in updated MALDI-TOF MS database of 2 manufacturers. Burka, 12 strain, and Biomeru, 8 strain. And, this means increase in the database of strains, species causing food spoilage results in increase of agreement rate of identification. The key to success so far was to design an elaborate framework of the technique and to network various stakeholders. Project members include 14 companies in food industry in Japan, more than 20 expert members. Current status: we finished the feasibility study of application of new methodologies to identification of food microbes. However, mold identification by MALDI-TOF MS is still unstable and difficult. We have started collaborative investigation of the methodologies with NITE Biological Resource Center, NBRC. NBRC is a National Research Institute of Japan. Why mold identification is unstable? For example, this is different spectra from same colony. Probably this spectra is from spore or from filamentous region. And different growth rate from different species. This is a view of stable identification by MALDI-TOF MS. Bacteria is excellent to fair. Yeast is very good. Mold, fair to poor. We are investigating improved methodologies using some typical food molds for rapid identification. Last slide. Expected outcome. MALDI-TOF MS technique will drastically speed up the microbe identification to ensure the microbiological food safety of products and to solve microbiological problems quickly. Increase of agreement rate of identification for spoilage bacteria results in the same above. All stakeholders in the food industry will share the information on the new technique with each other, which will surely strengthen the food industry and benefit the consumers. Thank you for your attention.
We have some time for a couple of questions, so I would like to see if there is any question right now. You can come up with to the microphone. Thank you.
Very nice presentation. I'm Mike Doyle from the University of Georgia in the U.S. Can you tell us how many cells you approximately need in order for the MALDI-TOF to do the analysis? How many of the bacteria cells
細胞の数。セルナンバー？sorry, 1億って言ったら、100 billion cells we need.
Okay, thank you. So it's not rapid, and you can't test it immediately from the product unless it's highly spoiled. Secondly, you say for bacteria, the range is excellent to fair. So what percentage of the bacteria fall in the fair range versus the excellent range?
ちょっと助けてください。Fair means over 90% accuracy. Okay. Almost all standard strains, but wild type is difficult sometimes.
Okay, thank you.
Nancy Bontempo from Mondelez. Kind of to Mike's question, on one of your first slides, you had a picture of a sample slide. Was that supposedly a petri dish, or so if you have a petri dish with all different variety of microorganisms, you're going to be able to point this laser at that colony and get the identification? Is that correct? No, okay.
すみません、教えてください。
So could you explain the sample preparation a little bit? Yes, thank you.
Sample preparation?
Yeah.
Sample preparation. Sample in smear method, only pick up raw colony to stainless steel. And stainless steel. Only pick up and matrix solution and irradiation. Very easy technique.
I'm not asking a question, but I just want to explain because I see the language difficulty. Basically, in addition to what you are saying, the method of preparation is use a toothpick, but you need a pure culture. So you simply take a toothpick on the edge, very minute quantities. You pick on the colony for bacteria, and then you smear you put on a metal sort of a metal plate. And then after putting on the metal plate, you put the matrix, and then you let it dry. After that, you put on the MALDI. But actually, so there's not much preparation from that angle. But one can follow the route where you have to do an extraction. Extraction, that is usually what is mostly used for molds. Because to directly do for molds is quite difficult, but you have to extract the ribosomal proteins, and then you prepare along those lines. And as he says again, for bacteria it's quite accurate in the sense that the profiles that you get, the peaks that you get are quite consistent, and to identify an unknown belonging to a specific group, it's quite high. But for wild type that are not in the system, it is difficult. But the good thing about MALDI, as we have used it from our side, is that when you come across that which you don't know, you just do your about 12 replicates, and then you create your own database in your own lab, and you identify against this unknown, although it is not within Biomurex or the other databases. You can create your own database within the lab and identify with such high accuracy. Thank you.
Thank you. Thank you.